Assalamualaikum and good afternoon everyone. Today we will learn about the histology of basic tissues 1 and 2. I'm sure you still remember the basic tissues 1 and 2 in your anatomy part during your semester 1. And today we will learn on the histology part. Okay, let's begin. For the introduction, you know already what is histology. It is a study of tissues. And there are 4 major tissue types in human body. But for this topic, we will focus on tissues 1 and 2, which are the epithelium tissue and connective tissue. For the epithelium tissue, you also know that there are 2 types, the surface epitheliums and glandular epitheliums. And for the glands, there are exocrine and endocrine. And for the surface epitheliums, you will name the epithelium according to the number of layers and cell shapes. Okay. For the layers, it is either simple, which is single layer, or stratified, which is more than one layer or several layers. And for the cell shapes, it is either squamous, which is thin and flat. And second, cuboidal, which is in a cube shape. And third, columnar, which is in a column shape. Alright, and these are all the pool images of simple, squamous until stratified, columnar. Okay, these are all the basic knowledge that you learn already in the semester 1. Okay, because we want to focus on the histology part. Okay, that's why I refresh back on the basic knowledge. Alright, we will start first with simple squamous epithelium. It is a single layer of thin, flat cells and has flat nucleus. Example location, okay, I'm sure you know already the location of simple squamous epithelium. Here I put some examples. And for the name, the terminology, if lining the ventral body cavity, it is called mesothelium. And the terminology for the epithelium that lines the heart and blood vessel, it is called endothelium. Alright, here is a histology image of the simple squamous epithelium. Alright, example location here is in kidney. Alright, this is from our own faculty slide box, slide F1, kidney. And if we zoom, this is an zoom image, high magnification image. Okay, I put here the 2 arrows showing you the specific location of the simple squamous epithelium lining the Bowman's capsule. Okay. Second, stratified squamous epithelium. There are 2 types. Okay, you still remember there are 2 types. First, the keratinized, and second, non-keratinized stratified squamous. Okay, and the locations are also different. Okay, between keratinized and non-keratinized stratified squamous epithelium. Alright, here is the histology image of the non-keratinized stratified squamous. Okay, non-keratinized meaning that there is no keratin layers, okay, on top of the stratified squamous epithelium. Okay, as shown here in this bracket, these are all the structure of the non-keratinized stratified squamous epithelium on the left image. And on the right image, I have zoomed in, okay, in the high magnification image of the stratified squamous. You can see that the single layer consists of the squamous cells, okay, but it stacks together, okay, in several layers, become stratified squamous epithelium. Okay. Example location here is in the esophagus. And second one is the keratinized stratified squamous epithelium. Okay, so the difference between this image and the previous one is the keratin layers, okay, as shown here in the a small green color arrow. Okay, that is the keratin layers. Okay, but then the specification is similar. They have stratified squamous epithelium, only that they have the keratin layers on top of the stratified squamous epithelium. Alright. Here as shown in the black arrow, it is the keratinized stratified squamous epithelium, and in high magnification image on the right side you can see the big image of the stratified squamous epithelium. Alright. Example location is in the epidermis of the skin. Alright, after that it is a cuboidal epithelium, okay, the third epithelium. So again in cuboidal also we have simple cuboidal and stratified cuboidal, and the locations are also different and the structure are also different. Okay. Okay, let's look first at simple cuboidal epithelium. Okay, example here is in kidney. Okay, this is in kidney tubules. Okay, you can see the ring shape here all consist of the single layer of cuboid shape of cells. Okay, compared to this is the example location in thick skin. Alright, in the sweat glands. Okay, earlier you see only single layer of the cuboid shape of cells, but here you can see more than one layers. Okay, it's stacked together. Alright, the cuboid shape of cells have more than one layers. Alright, you can see it from the nucleus. Alright, here is a cartoon image of the simple cuboidal and simple and stratified cuboidal epithelium. Alright, to differentiate, simple cuboidal only have one layer of cuboid shape of cells. Okay, compared to the stratified cuboidal, you have more than one lining. Okay, you see the nucleus also stacked together. Okay, they have two layers here, so more than one layers already called stratified cuboidal epithelium. Alright. Next is the columnar epithelium. Okay, again we have simple columnar and stratified columnar epithelium, and the locations are also different. Okay, let's start first with simple columnar. The example location here is in the duodenum, alright, in the small intestine. As you can see first in the left image, okay, that is a lower magnification of the simple columnar epithelium, okay, in small intestine, to be specific in the duodenum. But if you zoom the image, as you can see from the right image, okay, it is a high magnification image of the simple columnar. There is a one single cell which is in a column shape, right? Long and tall column shapes. Okay, it is simple columnar epithelium because it is just one layer. Okay, and then we have stratified columnar epithelium. Okay, the example location here is in the eye. Okay, conjunctiva of the eye. Alright, although the name is stratified columnar epithelium, yes, it should have a column shape of cells in several layers. But in real life, actually, in the real image, as you can see here, only the top layer is in a column shape, but the next layer, second and third layer, it's more like a cuboid shape of cells. Okay, because it packs together. Alright, but we still call it stratified columnar epithelium because the top layer is in column shape. Okay, and have several layers. And then we have two more additional epithelium, which are transitional epithelium and pseudo-stratified columnar epithelium. Okay. Alright, here is the image of the transitional epithelium. Example location in the urinary bladder. Okay. Transitional epithelium is a stratified tissue. Okay, it is made of multiple cell layers, as you can see here, and it has the ability to change the shape. Okay, when the bladder is in a relaxed state, okay, the cells is in a cuboidal shape. But when the urinary bladder is expanding, okay, it's full and it expands, the cells will also become flattened, okay, like a shape of simple squamous. So if you see here in the image here, there are in a cuboid shape. So the bladder here is in a relaxed mode. Okay. And we have also here image of pseudo-stratified columnar epithelium. Okay. The example location is in the respiratory tract, which is in the trachea. Alright. Actually, it's just one layer, but it has a pseudo image that you make it seems like it has several layers. Okay. And it has a cilia, okay, in trachea. So the name is ciliated pseudo-stratified columnar epithelium. So to recap, can you please answer this question? Under the light microscope, which type of epithelium can be found in the skin surface? Okay, I just talked about it just now. Second question, identify the epithelium that lines the urinary bladder and changes in appearance as stretching occurs. Okay, I also just mentioned about it just now. And the third question is why a pseudo-stratified columnar epithelium is not truly stratified. Okay, write down your answer in the comment section below. Alright students, we have finished with the surface epithelium. Now we move on to the glandular epithelium. Okay, I'm sure you know already we have endocrine and exocrine. Okay, the difference is the duct, the appearance of the duct. But for this histology part, we will learn the histology of the glands according to the type of the secretory products of the glands. Okay. Alright, there are three types of secretory products of the glands. First, it is serous, means clear or watery solution. Second, mucous, which is thick and viscous solution. Or it can be also a mix between serous and mucus. Okay, and in some books they put it as seromucous. Alright. Alright, first is the serous gland. Example location such as in the parotid gland or in the pancreas. Okay, and what is the characteristic of serous gland? Okay, first it is composed of acini with narrow lumen. Okay, as you can see from the image below, the black arrows, the single of black arrows showing you a single unit of the serous acini or serous gland. Okay, and you can't really see the lumen. Okay, lumen, which is the hollow structure, is very narrow. Okay, and that is the first characteristic of serous gland, narrow lumen. Second, it has secretory cells, which is round and basally located nuclei or nucleus. Okay. Example here in the small green arrow. Okay, it is one serous acini which has a round basally located nucleus. Okay. And the third characteristic is the cytoplasm of the cells is basophilic. Okay, basophilic is just a terminology, means that basophilic, basic liking, okay, means bind to basic dye. Okay, example of basic dye is hematoxylin. Okay, it will give a purplish color, right? Second gland is the mucous gland. Okay, example location in the esophageal gland. So what are the characteristics of mucous gland? Okay, as compared to the serous gland, for mucous gland, it has white lumen. Okay, so here in the two images here, you can see the label lumen. Okay, so and lumen, which is the hollow structure, you can clearly see the lumen now because it is really wide compared to serous gland, which is very narrow. Okay, second, the secretory cells now have flattened nuclei at the basal. Alright, it is very flat, okay, compared to serous, which is round. And the third one is the cytoplasm is very weakly stained, okay, very pale, okay, pale appearance, okay, weakly stained. Okay, remember these three characteristics. Alright, how about the mix of both serous and mucous, which is seromucous gland? Okay, example location such as in the salivary gland or in the submandibular gland. So of course, because it is mix of serous and mucous, it has both structure, which is mucous acini. Okay, as you can see here in the orange arrows, okay, it is the mucous acini. Okay, and it is surrounded by the serous cells, okay, which is shown here in the blue arrows. Okay, first the mucous acini, and it is surrounded by serous cells. Okay, okay, we have done the epithelial tissue. Now we move on to the second tissue, which is the connective tissue. Okay, you still remember we have several connective tissue types, okay, which are the connective tissue proper, which consists of loose and dense, and then we have supporting connective tissues, which have cartilage and bone, and we also have fluid connective tissues, okay, which are blood and lymph. And same again, the components of connective tissue proper, okay, cells, fiber, and ground substance. Here are the several images of the cells in the connective tissue, okay, fibroblast, plasma cell, adipose cell, large lymphocyte, macrophage, fibrocyte, eosinophil, neutrophil, okay, cells with pigment granules, small lymphocyte, and mast cell. Okay, we want to focus first on the connective tissue proper. Okay, the loose connective tissue, areola, reticular, and adipose tissue. First, for the areola tissue, okay, this is from our own slide box, slide A6. Okay, it is part of the loose connective tissue, okay, because it has more ground substance and less fibers. And in areola tissue, we have all the three types of fibers: collagen fibers, elastic fibers, and reticular fibers. Okay. And in the reticular tissue, we only have the reticular fibers. Okay. And adipose tissue, we have the adipose cells. Okay. Example location for the adipose tissue, it is in the subcutaneous layer of the skin. Okay. And For the dense connective tissue, we have dense regular and dense irregular. Okay. First, the left image is the dense regular, okay, such as in the tendons, ligaments, okay. Okay, the connective tissue is well organized, okay, in a regular manner, okay, and very packed, okay, with fibers, with collagen fibers. But for the dense irregular connective tissue, it has a disorganized structure of the fibers. Okay. Example location for dense irregular connective tissue, such as in the superficial layers of bones, cartilage, and peripheral nerves, and also in the dermis. Okay, here I show you the high magnification image of dense regular and dense irregular. Okay, for the left image, dense regular, example location is in the tendon. Okay, it has a very packed fibers, but it is arranged in a well organized manner. Compared to dense irregular, it has thick fibers, but it is arranged, it is disorganized. Okay, and the example location is in the skin. Next is the cartilage. Okay, cartilage has the chondrocytes, which is the cartilage cell, and chondrocytes are in the sitting in the lacuna, which is in the cavities in the matrix. Okay, so we have three types of cartilage: hyaline, elastic, and fibrocartilage, or in some books, fibrous cartilage. Okay, first hyaline cartilage. For hyaline cartilage, it has a glassy appearance. Okay, and as you can see in the arrow here, it is a chondrocyte. Okay, sitting in a lacuna, and the surrounding structure of the chondrocyte is the ground substance. Okay, it is very clear. Okay, so that is the characteristic of hyaline cartilage. Okay, very simple to distinguish. And the second cartilage is elastic cartilage. Okay, also similar with hyaline cartilage. It has a chondrocyte, okay, sitting in a lacuna, but the surrounding structure of the chondrocyte, it has elastic fibers. Okay, that is the difference between that makes elastic cartilage different to other cartilages. Okay. And the third cartilage is the fibrocartilage. Okay. Here in the histology image, okay, the characteristic of fibrocartilage, it has rows of chondrocytes. Okay, the chondrocytes are sitting in a row together. Okay, so this is also different compared to elastic and the hyaline cartilage. Okay, so where the chondrocytes are dispersed compared to this fibrocartilage, the chondrocytes are sitting together in a row. Okay, and the surrounding of the chondrocyte, it is packed with the collagen fibers. Alright, next connective tissue is the bone. Okay, this is slide A10, from our own slide box, the image of bone. Okay, and the left image, okay, you can see the labeling of spongy bone and compact bone. Okay, so under higher magnification, okay, let's focus first on the top box image. Okay, so we have the lacuna. Alright, the osteocytes are sitting in the lacuna, in the bone. Okay, and the peripheral structure of the lacuna is called canaliculi. the branching structure, okay, from the lacuna. It is called canaliculi. And the bigger image here is you can see the section of compact bone. Okay, why it is in a black color? Because we use a silver staining, okay, not HNE. Alright, in the bigger image, first labeling you can see Volkmann's canal, okay, and the second labeling is the Haversian canal, okay, and the third labeling is the lacuna, okay. So the lacuna, the zoom image is in the top small box, okay. Here is another cross section of the bone, and this time it is a spongy bone. Okay, this is also from our own slide box here in the faculty, slide A10 or A11. So in the spongy bone here, in the center part, you can see there are a lot of bone marrow. Okay, and at the edge of the bone marrow, you can see there is a trabecula. Okay. Okay, let's zoom the image of the trabecula. Okay, when you zoom the image of the trabecula, okay, it is surrounded by a lot of bone marrows. Okay, in the marrow space there are a lot of bone marrows. Okay, so first we have in the bone matrix or in the trabecula, all right, we have the osteocytes which are sitting in the lacuna. Okay, osteocyte means the bone cells. Okay, we have osteocytes and osteoblasts, but osteocytes are sitting in the lacuna in the bone matrix, while osteoblasts are sitting in the periphery, okay, of the bone matrix. And then for the blood, I will not cover much on the blood, just a touch on it, because you will learn more about the blood structure in your second year. So we have erythrocytes, leukocytes, and platelets as a component in the blood. And leukocytes, we have several components: neutrophils, eosinophils, basophils, monocytes, and lymphocytes. Okay, first with the erythrocytes. Okay, this is the erythrocytes as shown here in the labeling. And the small structure in the second labeling, it is called platelets. Okay, you can see the difference and the comparison of in size between erythrocytes and platelets, okay, from this one image. Alright, here is the white blood cells. Alright, and in white blood cells compared to red blood cells, it has a nucleus. Okay, so the components in white blood cells are neutrophils, eosinophils, basophils, monocytes, and lymphocytes. Okay, you know already neutrophils, the structure is easily to distinguish. Okay, and monocytes and also lymphocytes easy to distinguish due to the shape of the nucleus. But and then how are you going to distinguish your eosinophil and basophils? Okay, because both have granules in the cytoplasm. But easy to distinguish because eosinophil here gives the pink color, okay, pink or orange color, but for basophil it gives the purplish color, right? Here's the summary of the blood. Okay, I think I have mentioned this in the first semester. Okay, this is just a basic structure of each component of the blood cell. With that, I think that's all for the histology of basic tissues 1 and 2. If you have any further question, you can email me or just drop a message in the UKM folio or in the Microsoft Teams. Okay, thank you everyone.